Welcome back. Today I will take a class about tissue culture technology. You know that tissue culture technology is a part of biotechnology. Today we'll discuss briefly about tissue culture system, or you can say that tissue culture process or steps and its uses or applications, and how tissue culture processes contribute to conserve our natural resources or biodiversity. It is also important part about today's discussion. So hope you will enjoy this class very much. Okay, my dear student, let's start. Today's topic is tissue culture. Before define tissue culture, we have to know about the biotechnology. You know that biotechnology is a elaborate science of biology involving living system and organisms to develop or make product. Depending on the tools and applications, it often overlaps with related scientific field, such as tissue culture is one of them. So you know that in the late 20 and early 21st century, biotechnology has expanded to include new and diverse. According to your book, we can define biotechnology a wide range of produces for modifying living organism according to the human purposes, going back to demonstrate of animal cultivation of the plants and improving to these though breeding programs that employ artificial selection and hybridization. Biotechnology as the application of biological organism systems or processes by various industries to learning about the science of life and the improvement of the value of materials and organisms, such as pharmaceuticals, crops and livestock, livestock, etc. Biotechnology is the integration of natural science and organisms, cells, parts and molecular products and services. In broad sense, biotechnology is the research and development in the laboratory using bioinformatics for exploration, extraction and exploitation and production from any living organisms and any sources of biomass by means of biochemical engineering, where high value added product could be plant, for example you can say biosynthesis, reproduced by biosynthesis, etc. So now we'll discuss about the tissue cultures. So first I want to explain what is tissue culture. Plant tissue culture is a collection of techniques used to maintain or grow plant cell, tissue or organ under the sterile condition on a nutrient culture medium of known as composition. It is widely used to produce clones of a plant in a method known as micropropagation. Micro mean that small particles, small tissue. Propagation is a inner capability to produce a adult plant. Different techniques in the plant tissue culture may offer certain advantages over traditional methods of propagation. So in broad sense, the production of exact copies of plant that produce particularly good flowers, fruits or have other desirable traits, to quickly produce mature plant, the production of multiples of plants in the absence of seeds or necessary pollinators to produce seeds, and after next the regeneration of whole plants from plant cells that have been genetically modified. The production of plant in sterile containers that allows them to be moved with greatly reduced changes of transmitting disease, pest, and pathogens. Also, you can add the production of plants from seeds that otherwise have very low chances of germination and growing. For example, orchid is one of them. And furthermore, to clean particular plant of viral and other infections, and to quick multiply these plant as clean stock for horticulture and agriculture. So in industry like horticulture and agriculture, here tissue culture is very favorite technology. Plant tissue culture relies on the fact that many plant cells have the ability to regenerate a whole plant. It is called totipotency. So totipotency is a major term when you want to explain tissue culture. Single cell plant cell without cell wall, pieces of leaf, stem or root can often be used to generate a new plant on culture medium given the required nutrient and plant hormone. So together required nutrients mean that glucose, fructose, water, and plant hormone mean that you know that different kinds of plant hormone present in nature, like that auxin, gibberellin, etheriline, these are phytohormone. So together you can say that culture medium. Culture medium totally depend on the laboratory. Although some growers and nurseries have their own labs for propagating plants by the technique of tissue culture. A number of independent laboratories provide customs propagation services. The plant tissue culture information exchange leads many commercial tissue culture lab. Since plant tissue culture is a very labor intensive process, I think this would be an important factors in determining which plants would be commercially viable to propagate in a laboratory. So now I want to explain the techniques. So what are the steps of tissue culture? First of all, you have to select a mother plant and have to exact explant. The tissue obtained from a plant to be cultured is called an explant. Explant can be taken from many different parts of a plant, including proportions of shoots, leaves, stems, flowers, roots or single undifferentiated cells, and from many types of mature cells, provided they still contain living cytoplasm and nuclei and are able to de-differentiate and resume cell division, you can say. This has given rise to the concept of totipotency of plant cell. However, this is not true for all cell or for all plants. In many species, explants of various organs vary in their rates of growing and regeneration, while some do not grow at all. So at the very starting, you have to make culture medium. Preparation of tissue for tissue culture is performed under a aseptic condition under the, you can say, filtered air provided by a laminar flow cabinet. Thereafter, the tissue is grown in the sterile containers such as petri dish or flask in a grow room with controlled temperature and light intensity. Living plant materials from the environment are naturally contaminated on their surface with microorganisms, so their surface is sterile in the chemical solution. Before suitable sample are taken, the sterile explant are then usually placed on the surface of a sterile solid culture medium, but are sometimes placed directly into a sterile liquid medium. Particularly when cell suspension cultured are desired, solid and liquid medium are generally composed of inorganic salt plus a few organic nutrients, vitamins and minerals hormone. but solid media are prepared for the liquid medium with the addition of a glying agent, usually purified agar. The composition of medium, particularly the plant hormones and the nitrogen sources, have profound effect on the morphology of the tissue that grow from their initial explant. For example, an excess of auxin will often result in a proliferation of root, while an excess of cytokinin may yield shoot. A balance of the auxin of both auxin and cytokinin will often produce an you can say unorganized growth of cell or callus. But the morphology of the outgrowth will depend on the plant species as well as the medium of composition. As culture grow, piece of typical sliced of subcultured on new medium to allow for the grow or to alter the morphology of the culture. I think the skill and experience of the tissue culturists are important in judging which species to culture and which to discard. It depend on total experience. As shoot emerge from a culture, they may be sliced off and rooted with auxin to produce plantlets, which mean mature can be transferred to potting soil for further growing in the greenhouse as a normal plant. So this is the important point. The tissue obtained from a plant to be cultured is called explant. Previously I mentioned. But the specific difference in the regeneration potential of different organs and explants have various explanations. The significance factors including differences in the stage of cell in the cell cycle, the ability of transport endogenous growth regulates, and their metabolism capability of the cell. The most commonly used tissue explant at the meristematic end of the plant, like the stem tip, auxiliary bud tip, and the root tip. These tissue have high rate of cell division and either concentrate or produce required growth regulation substance, including the auxin and cytokinin. Shoot regeneration efficiency in tissue culture is usually quantitative trait that often varies between the plant species and within a plant species among the subspecies, varieties, cultivars, or ecotypes. Therefore, tissue culture regenerate can become complicated, especially when many regeneration products have to be developed for different genotypes within the same species. Three common pathways of plant tissue culture regeneration and propagation from pre-existing meristem, organogenesis, and non-zygotic embryogenesis. So you will get more idea about these types of term in a higher class, I think. The propagation of shoot and node segment is usually performed in 4 stages for massive production of plants through the in vitro vegetation multiplication, but organogenesis is common method of micropropagation that involves tissue regeneration of adventitious organs or auxiliary buds, directly or indirectly, you can mention. So now we'll discuss about the benefits of tissue culture. So you know that to conserve endangered species, tissue culture is very effective. So if I discuss according to your syllabus, plant tissue culture is used widely in the plant sciences, forestry, and horticulture application includes. The commercial production of plant used well as potting, landscape, and florist subject, which uses meristem and shoot culture to produce large number of identical individuals. This is very important. It helps to produce large number of identical individuals in sense of commercial production, you can mention. Secondly, the conserve rare or endangered plant species. You know that Nema bengalensis, in Bengali we can say that tali palm, it's a endangered plant. So if you want to conserve such kind of plant, tissue culture is vital technology. Number next, a plant breeder may use tissue culture to screen cell rather than plant for advantageous character, example herbicide resistance or tolerance. So by using this method, we can minimize use of different kinds of insecticides and pesticides. It is very effective for our environment. And after next, you can say large scale growth of plant cell in liquid culture in biofactors or bioreactors for production of the valuable compounds like plant derived secondary metabolism and recombinant protein used as you can say biopharmaceuticals. So biopharmaceuticals also depend on the tissue culture, you can say. You can also add to cross distantly related species by protoplast fission. You can say mention like hybrid and hybrid. You will get elaborate idea about the hybrid and hybrid and protoplasm fission and regeneration of the novel hybrid. It also possible using tissue culture. And to rapid study the molecular basis for the physiological, biochemicals and reproductive mechanism in plant, for example in vitro selection for stress tolerance plant. So it is very important. Even you know that some desert plant, if you want to culture in a moisture weather, now it is possible. You can use tissue culture technique and you can change their variety. And after next, to cross pollination also possible, and by this for chromosome doubling and induction of polyploid, for example double haploid, tetraploid and other form of polyploids. This is usually achieved by the application of tissue culture. It is very well known at this moment. And finally, you can say that synthetic seed, a somatic embryo, is a very important part of artificial endosperm and artificial seed coat. And lastly, I can mention production of identical sterile hybrid species can be obtained by the tissue culture. So that's all about the tissue culture. First of all, we discussed about the tissue culture process, definition and everything. Hope you will understand. Thank you very much for watching this video till end. And if you have any question, please put on comment box. Thank you again.